Very good morning, dear students. Today we are going to discuss the next important topic in unit number 5, biodegradation of petroleum hydrocarbons. And in the syllabus they have mentioned petroleum hydrocarbons, their microbial degradation. And one of the most important problem in the present scenario, the crude oil degradation. And the crude oil contamination, especially in the aquatic body, that create lot of problem to the ecosystem. It prevent the photosynthetic activities, and similarly it creates lot of problems in the aquatic ecosystem. Because of that particular issue, lot of organisms they have totally it has adversely affect their existence due to the oil contamination, and this we can see across the world, and this is the potential problem in the world. And microbiology, especially environmental microbiology, is going to play a very important role for the degradation of such kind of crude oil based petroleum hydrocarbon. So we are going to discuss, before we are discussing the details about the degradation aspects, so you should know what are petroleum hydrocarbon. Petroleum hydrocarbons are the complex substance formed from the combination of hydrogen, carbon. In addition to these two, the other elements such as oxygen, sulfur and nitrogen, and the sulfur content create an important part in the crude oil, and some may combination and some may be the combinations of various of these above elements, and they exist in various types also. And majority of the petroleum products are used in the automobile industry, combustion engine, to natural gas, you are knowing that, okay, and heating purpose and the cooking purpose. You are very commonly LPG, okay, liquefied petroleum gas we are commonly using. And this, there are a lot of informations are available about the crude oil and the the petrol, diesel and all other petroleum products. And you know that we are not going to discuss all those details. We are going to just summarize the introductory concept. And some oils are known as light oil, that depending upon the composition of the so-called important elements, and some oils are the known as heavy oils, depending upon the high concentration of all those elements, especially sulfur, okay. And some are wet gas and some are dry gas, depending upon the composition. And the major source, and these oil products are commonly present in the ecosystem and from the very ancient time onwards, okay. And the major source are the rock particle that is commonly present in the earth, okay, the source rock, okay. And the source rocks are the rocks that produces the crude oil petroleum hydrocarbon, and where they are very rich in particulate organic material, okay, and they are very particular in nature. As I said, the carbon and hydrogen along with the oxygen and sulfur, they forms various combinations, and there will be chemical changes after the burial and convert the plant and animal tissues, okay. Once the death of the animal and plants, so it will be released to the soil, and there it forms the complex molecule that produce oil or gas, okay, natural gas, okay. And you know that the importance of methanogens for this particular purpose, methane produces, okay. And by the effect of heat and pressure on various, these components will be trapped in the soil sediment beneath the earth surface over millions of years, and they produces the petroleum hydrocarbons. Okay, so that is the origin of petroleum hydrocarbons. Okay, then these petroleum hydrocarbons are commonly classified in different groups. So the classification of petroleum hydrocarbons, they are mainly depending upon the number of carbon atoms. Okay, the petroleum hydrocarbons are broadly classified into aliphatic petroleum hydrocarbons and aromatic petroleum hydrocarbons. And the aliphatic components are mainly classified into three groups: alkanes, cycloalkanes, and alkenes. Okay, so these alkanes, they contain single bonds between carbon atom. You know that. Examples are hexane, heptane, and octane. Okay, all these components are aliphatic petroleum hydrocarbon. Cycloalkanes, they contain carbon atom in a cyclic and ring form. Okay, the 5-membered or 6-membered ring form. Okay, methyl cyclopentane and ethyl cyclohexane are the examples of cycloalkanes. They are also important class of petroleum hydrocarbons. The next one is the alkene. They contain one or more carbon compound with a double bonds, and here the first two cases they are stabilized by single bonds, and here it is stabilized by double bond. Example, ethene and propene. Okay, these are aliphatic hydrocarbons. So this is all about aliphatic hydrocarbon component that is commonly available in the form of petroleum products. Next one is aromatic components. They are having an aromatic ring in very common. Okay, so that is, there are two types. One is monoaromatic hydrocarbons. Next one is polyaromatic hydrocarbon. Monoaromatic hydrocarbons are the hydrocarbon that contain only one benzene ring as a part of their structure. Okay, as a part of their structure that consisting of only one benzene ring. Example, BTEX. Okay, that is butane, toluene, ethyl, ethylene, then xylene. Okay, so BTEX. Okay, very common example. Next one is polyaromatics. They contain two or more fused or condensed benzene ring system. Okay, so for example, polyaromatic hydrocarbon. We have already discussed about polyaromatic hydrocarbon in the beginning of this particular unit. So these are the classification of petroleum hydrocarbons. Then if you look at the composition of crude oil, especially what are the major components of crude oil? Okay, that consisting of aliphatic hydrocarbons, as I said, aromatic hydrocarbons, then heterocyclic components. So aliphatic hydrocarbons are, they are N-alkane, that is example N-heptadecane. Similarly, isoalkane, here you can see branched, that is known as the pristane. Then cycloalkane, that is a cyclopentane or cyclohexane. Okay, so these are aliphatic hydrocarbon. Then aromatic hydrocarbon, toluene, you know that is the structure of toluene, and their aromatic ring system. All the things you can see the ring system, that is the main important thing. 6-membered ring system: naphthalene, pyrene, phenanthrene, and acrysene. Okay, these all are the then benzopyrene. Okay, these all are the aromatic hydrocarbons. Then heterocyclic compounds are their nitrogen containing heterocyclic compound, that is carbazole. Then sulfur containing heterocyclic compound, that is dibenzothiophene, one of the major important pollutant in the ecosystem. Then oxygen containing heterocyclic component, that is xanthenes. Okay, so these are the heterocyclic components, and these are the overall chemical composition and mixture of the crude oil or petroleum hydrocarbons. Then what are the major approaches for the degradation of petroleum hydrocarbon? PHC, throughout the presentation I will use PHC, that is petroleum hydrocarbon, that is short form. Okay, so PHC degradation are, as we have discussed in the previous topic, there are three important approach. One is chemical methods, next one is the physical methods, next one is the biological approach. The chemical method, this petroleum hydrocarbons are commonly, that is directly we are injected to the chemical, injection of chemical oxidants to the contaminated and open soil, there it will be contaminated. Then second one is the release of this components directly to the groundwater. These are highly dangerous approaches that totally deteriorate the ecosystem balance, the ecological balance, and it contaminates the biogeochemical cycles as well. Then this is not at all an ideal method. Then the physical method, they are the disposal in the form of landfill. Okay, it requires a lot of lands, and that is not an excellent method that we have already discussed. Next one is incineration. Okay, incineration and pyrolysis, such kind of processes are very common physical process. That is also not a excellent method because it has lot of shortfalls. Not only that, the degradation will not happen effectively. Then we are going to focus on the microbial, especially biological approaches. The biological process, there are mainly two types. One is plant-based process, that remediation, okay, remediating such kind of components are known as phytoremediation. Then the microbial based processes or bioremediation. Okay, so we are going to discuss the microbial based processes or bioremediation of the oil space. Okay, so we are going to discuss how the contaminants are degrading in the oil spills by microbial origin. Okay, so then there are so many general factors which influences the common degradation of petroleum hydrocarbons. Okay, the main factors which impacting the petroleum hydrocarbons degradation are the nutrients. Okay, then the microbial community in which that is present, the toxicity of this particular petroleum hydrocarbons, the activity of water. I told it is commonly present in the water ecosystem. Okay, so the bioavailability, then the hydrogen ion concentration, the pH, okay, the salinity, salt concentration, and the oxygen, okay, the presence of and absence of oxygen, aerobic and anaerobic condition, then the temperature. So these are the major general factors that influences the degradation of petroleum hydrocarbons. Then if you look in microbial aspects, the general aspects, there are specific factors which influences the degradation of petroleum hydrocarbons. Okay, these hydrocarbon susceptibility to biodegradations are various parameters. One is the microbial properties. The main factor is the microbial properties. Next one is the environmental properties, and third one is the hydrocarbon properties. Okay, if you look at the microbial property, there is a regulation of gene, because you know that the degradation of the hydrocarbons are happening due to the certain specific enzymes. That enzymes are protein that you know that is obtained from the transcription and translation of the gene. Okay, so that is the gene regulation, and the surface hydrophobicity, then the metabolic flexibility. Most of the biodegradation processes are the metabolic processes. Okay, and that is the uptake mechanism, how they are taking this particular component, toxic components into the cell, and the tolerance to the toxicity, how much tolerable these toxins are. Okay, how much they are able to tolerate. Okay, this is the microbial features. Then the environmental properties are the electron acceptors, the presence of nutrients, and as we have seen in the previous slide, that is the temperature, salt concentration, salinity, and pH. Okay, majority of the cases that is happening in the marine and aquatic ecosystem, the freshwater and brackish water and marine ecosystem, there will be lot of such a kind of parameters are available. Then the important parameters, the nature of the hydrocarbons. Okay, their bioavailability, the molecular structures, whether it is aliphatic or whether it is aromatic. Okay, whether it is alkane or alkyne or alkenes. Okay, then their toxicity and their physical state. Okay, and how they are interacted and condensed. Okay, and what are the chemical composition, they are present only hydrogen, only carbon or sulfur. Okay, nitrogen, oxygen, all those kind of specialities are also important parameter for the degradation of hydrocarbons. Then now we are going to talk about the microbial degradation of petroleum hydrocarbons. There we are going to discuss the aerobic degradation. So this is the general and simplest pathway of the microbial activity towards the petroleum hydrocarbon. So this is the hydrocarbon, okay, this is a microbial cell, you know that, okay, and initially this will be attacked by the enzyme oxygenases in presence of oxygen, and the degradation by the peripheral pathway, it will convert it into other pathway, and they will be converted into C2 compounds, and this C2 compounds will be converted combines with the C4 compounds, and it will be complete and form a C6 compounds, and it will be produce the C5 compounds, and this recycling will happen through TCA cycle, tricarboxylic acid cycle. You know that there is a transformation of the number of carbon atom. Tricarboxylic acid is the first intermediate that is present in the citric acid cycle. There will be having carbon dioxide, oxygen and water that will be reaching to the electron transport system, and it will be going to the respiration process. And similarly you will be having the biosynthesis, and it will be using ammonia, phosphate, sulfate, and ions, okay, ferric ion, okay. So that will be the cell biomass and growth. So what I am trying to tell is the hydrocarbon, the crude oil can be degraded by the petroleum hydrocarbons can be easily degraded by the microorganism by the activity of various types of oxygenases and other enzymes, okay. Then this is a specific example. Here you can see an alkane, how degradation of petroleum hydrocarbon, simple alkane, okay, aliphatic hydrocarbons are happening, okay. Here you can see alkane. So using here you can see oxygen monoxygenase enzymes, okay. This R CH2 CH3 is a simple alkane. Then what you can see is it will be oxygen will be added. So that will be, instead of one hydrogen, the hydrogen and the oxygen will go and bind, and it will produce a alcohol, okay. This is a secondary alcohol, and the release of 2H plus will happen. Here you can see the release of these two hydrogen, okay. Then you will be able to see there is a ketone, okay. First there will be an alcohol, then it will be a ketone will be produced, okay. So once the ketone is produced, then what will happen is it will be again oxygenation will happen, oxygenation and reduction, both the process will happen, okay. And you can see one more oxygen is added. There is, you can see C double bond to O and O, that is an ester linkage, okay. So this is an acetyl ester component, okay. So then the acetyl ester component, and what will happen, there will be a cleavage of this water will be added, okay, and this water will be dissociated at H plus plus OH minus, and that OH minus will be coming and binding, and you can see CH3 COOH will be released out, that in the form of acetic acid, and you can see that the CH2 or the other H that is coming from this H2O that will combines with the RCH2OH, that is primary alcohol, and this primary alcohol will be converted into aldehyde, okay. Here you can see again reduction will happen, this hydrogen, okay, and this hydrogen will be reduced, okay. Then we will be having RCHOH, okay, that is RCHO, okay. Then aldehyde you will be getting, okay. And once the aldehyde is produced again, the water will be released out. Then you can see what will happen is the carboxylic acid, oxygen will be again, you can see that H plus plus OH minus will be added. Here you can see hydrogen will be added, water will be released out, then one hydrogen will be added here, then you will be able to see COOH, okay, then RCOOH, okay. And here you are having an acid, and another here also you are having an acid. So once the acids are produced, you know that this acid will be undergoing, in the last class we have seen, okay, we have discussed about the degradation of LAS, okay. So that such a kind of components will be beta oxidation will be happening. So here also the beta oxidation will be happening, alkyl benzyl sulfonate, yesterday we have discussed, okay. So the acetic acid will be undergoing with a series of steps that will be undergoing beta oxidation, and again carboxylic acid will be undergoing beta oxidation, and will produce finally the end product actually. So that is the overview of the degradation. Then again another specific example is then N-alkane. Here you can see a monooxygenase reaction. How enzymes are playing a very important role. Enzymatic reactions involve the process of hydrocarbon degradation. That is what I told, monooxygenase reaction. Okay, so in presence of certain cofactor, actually that is rhodopsin. Okay, here it will be coming NADH converted into NADH, and N-alkane will be converted into primary alcohol. Okay, and once it is produced, it will be pass through these steps and completely it will be degraded. So this is about the aliphatic hydrocarbon. Then what about aromatic hydrocarbon? So here you can see benzene. The same monooxygenate enzymes will be activated here. Oxidation will happen. Then NADH will be reduced to NAD plus. Then you will be able to see arene oxide. Okay. Then water molecule will be added. Then the trans dihydrodiol will be produced. Here you can see H2H and 2 hydroxyl groups. Okay. Here you can see one oxygen is commonly sharing with this structure. Okay. Then again there is NAD, that is converted into this hydrogen. They will be accepted, and NADE plus will be converted into NADH. Then you can see that will be this H and this H will be degraded. Okay. Then you will be able to see a 6-membered ring plus this OH. This is known as a catechol. I told you most of the cases the catechol are the end product. Okay. So another important cases, this is monooxygenase, but here the dioxygenase also can be taken part in this particular case. So benzene, that dioxygenase, the oxygen will be diatomic, the oxygen will be added, that NADS, DH will be converted to NAD plus. Then in a single stage you can see that oxygen will be added. Okay, you can see that there is a positional difference. Okay, this is a transposition and this is cis position. Okay, this is cis conformation. Okay, and you can see the same thing, catechol will be produced. Catechol, once the catechol is produced, so what will happen is it will undergo other further metabolic pathway and to TCA cycle, and it will be completely degraded into the end products such as carbon dioxide, oxygen and water. Then what are I told about the enzymes. What are the major enzymes that is involved in the petroleum hydrocarbon degradation? The major enzymes and the major microorganisms are both bacteria and fungi are playing a very vital role. First one is soluble monooxygenase, soluble methane monooxygenase, that is generally acting on the C1 to C8 alkane and cycloalkanes. That is commonly all the methylococcus, methylocinus, methylocystis, methylomonas, okay, methylocella, all those organisms are having this methane monooxygenase. Then the particulative methane monooxygenase, this is soluble methane monooxygenase, and this is particulative methane monooxygenase. This is commonly present in their membrane. Okay, so this is commonly present in the cytoplasm. Okay, so here C1 to C5 halogenated alkanes and cycloalkane. So here you can see methylobacter, methylococcus and methylocystis. Okay, then you can see the ALKB related, that's a gene. Then alkane hydroxylases. Okay, C5 to C16 alkane and fatty acid, alkyl benzene, cycloalkane and so on. Okay, here you are the most of the cases the best organism is Pseudomonas. Okay, Pseudomonas, Bactodellera, then similarly Rhodococcus, Mycobacterium. Okay, these are the major organisms. Then in the case of this, all the things are prokaryotes, but in the case of eukaryotes, okay, eukaryotic cytochrome P450, that is C10 to C16 alkane fatty acid. There is a fungal yeast, okay, Candida maltosa and Candida tropicalis and Aerovina lipolytica. Okay, so these are the major fungal yeast. Then the case of bacterial for P450 oxygenase system is also there. There is C5 to C16 alkane, cycloalkanes are there, Acinetobacter, Colobacter and Mycobacterium are the major important player. Then all the things are either monooxygenase, hydroxylase, okay, and dioxygenase are also there. That's C10 to C30. That is the maximum number of alkane it can degrade. Okay, using the previous mechanism. So Acinetobacter species is an excellent example. So these are the major microorganisms. Again, there are many more. Some of the examples only I have shown here. So for your understanding. So these are the microbial enzymes in petroleum hydrocarbon that shown the high amount of degradation. Then another important aspects, in addition to microbial enzyme, in presence of certain biosurfactant, microorganism under certain specific condition, they can produce certain biosurfactant. There are recent studies revealed that the biosurfactant like rhamnolipids, okay, long chain lipid fatty acid, they produced by certain bacteria like Pseudomonas, they enhanced and accelerate the hydrocarbon uptake, okay, the hydrocarbon uptake by the bacteria and the degradation of this petroleum hydrocarbons, okay. So the bacterial cell will be produced the biosurfactant, and biosurfactant, this is a long chain biosurfactant. One end you can see a 6-membered ring, and you can see a long chain alkane here. Alkane here, there is a keto group, there is a carboxylic acid group, and that is known as rhamnolipids, okay. So this rhamnolipids, it's undergoing emulsification with the oil, okay. Then the emulsification with the oil, so that the oil will be taken up. The formation of the oil can be easily emulsified. There will be formation of micelles in the membrane of the bacteria, okay. That micelles, that is a fine fat globule that is combines with the that is the round small droplets of the lipid molecules that can be taken up by the cells, okay. And this micelles can be absorbed by the microbial system, and they can be engulfed, and they can degrade this component along with this micelles, okay. And the cell wall, it will be taken up to the cell wall and will be completely degraded by the cell. So what I am trying to tell is the role of biosurfactant are also important for the biodegradation of petroleum hydrocarbons or crude oil degradation. And there are so many limitations are there for conventional microbial degradation. Some of the limitations are, we cannot have a single microorganism for the degradation of all the organic waste, okay. No single microorganism can degrade all the toxic components. Next one is the high concentration of some of the organic components, especially I told you in the ocean, marine water, there will be lot of oil, okay, oil spills, okay. So that will be inhibit the activity of the growth of microorganism. So microorganism cannot survive in the extreme concentration of this particular oil. That can also happen. Then most contaminated sites contain a mixture of chemicals or organism that can degrade a very small number of chemical. Not all chemical can be degraded, okay. Only few chemical can be degraded, but the contamination will not be completely degraded, okay. That is another important problem. And several non-polar components that is commonly present in this petroleum hydrocarbon, that will be absorbed into the particulative matter in soil or sediment and become less available for the degradative microorganism. Instead of going to attracting to the microorganism, they will go and deposit along with the soil sediments at the bottom part of the ocean. So because of that particular reason, microorganisms are not getting this particular component for their degradation. Then the most importantly, this is microbial degradation, and the process is very slow, okay. Very big, based on the presence of functional group. We have already discussed the ortho para meta influence. If the functional group is in the meta position, how much time it will take to degrade, we have seen, okay, than the ortho and para position. So it is very important process. So it is very slow process, okay. That is another important thing. Then what is the development, okay, for this particular thing? There is a very interesting concept that is very important concept, this one, okay. So in order to solve that particular aspects, okay, only one single bacteria, then the question is, because of all these drawbacks, now scientific community thought that why can't we create a bacteria, they can degrade multiple hydrocarbons, okay. All the hydrocarbons are degraded by the presence of the gene. Okay, so that genes are the one it is producing the enzymes. Okay, so why can't we selectively identify those genes, and why can't we transfer this particular gene, and why can't we create a genetically engineered organism? And this particular, the first and foremost discovery in this particular area is made by an Indo-American microbiologist. This is very, very important at this point of 2020. His name is Dr. Anand Mohan Chakrabarty. Okay, so he is the first person. He constructed a bacteria that can called superbug, because now superbug means antimicrobial resistant organism. Earlier it is superbug means the bacteria, they can degrade multiple hydrocarbons. Single bacteria that has a multiple gene to degrade so many hydrocarbons. Okay, and he constructed by transferring the plasmid from different strain of a particular type of bacteria called Pseudomonas putida. Okay, Pseudomonas putida is a gram negative. You know that Pseudomonas gram negative, motile organism. Pseudomonas is oxidase positive. Okay, so that you are already knowing the biochemical characteristics. Okay, so Pseudomonas putida. Okay, and this is commonly known as oil eating bacteria. Okay, superbug. Okay, because one single microorganism, what he has done is this the organism, he identified various strains of Pseudomonas putida. They are able to degrade multiple hydrocarbons. Okay, and they are identified the individual gene, and this gene they have specifically identified and transferred to a single bacterium. Okay, and he identified that this single bacterium could degrade all the four types of, all the 4 or 5 or 3 or more than multiple type of hydrocarbon with the presence of this particular a gene. Okay, and that is manipulation by the transfer of the plasmid, and the gene that is he has integrated to plasmid by restriction enzymes. Okay, that particular gene he has inserted into a small plasmid, a cloning vector, you know that. Okay, cloning vehicle, and transferred to Pseudomonas putida. Okay, and this experiment he has done in 1917s. Okay, and that is a great discovery. You know that the great Indo-American microbiologist. You know that very recently, 10th July 2020, he passed away actually. Okay, so I think very recently you might have seen, okay, in the newspaper magazine 2 months ago, you might have seen that that person, he was just 82. He was 82 years old actually. Okay, that particular time. Okay, his contribution is very, very comparable thing. And another important thing, this is the first bacteria in the history of microbiology, man-made bacteria that is getting patent. Okay, the Supreme Court, US Supreme Court, they have given a patent to Anand Mohan Chakrabarty for the creation of a superbug. Okay, Pseudomonas putida, they are able to degrade multiple plasmid. Generally, the patents will not be given to microorganism. Okay, the patent means the privilege. Okay, that is the authority. Okay, the IP, the intellectual property. Okay, so the ownership. Okay, so generally it will not be given to bacteria, but he got the ownership of this particular bacteria. And what he has done is he identified 4 strain of Pseudomonas putida. Strain number 1, the speciality of that strain has a particular gene that can degrade a particular hydrocarbon component, a particular hydrocarbon called a camphor, one of the most potential dangerous component called a camphor. Okay, so that particular gene he has integrated to a plasmid, and a camphor degrading plasmid he has created. Second one, another strain of Pseudomonas, he identified that this particular organism has oct gene. That oct gene, that is responsible for the degradation of octane. Okay, and that particular gene along with the plasmid, octane degrading plasmid, he has identified from the second strain. Then the third strain, he has seen this particular bacteria, Pseudomonas putida, strain 3 has a XYL gene, that is xyl gene, that is a xylene degradation gene. Okay, that has got xylene degradation plasmid. Okay, The next one is the NAH gene, okay, NAH gene. NAH gene, that particular gene will be getting an ability to degrade naphthalene, okay. So that naphthalene also another important hydrocarbon. This camphor, octane, xylene and naphthalene are four potential hydrocarbon that is commonly present in the crude oil, okay. And what he has identified, individual strain he has identified and taken the gene, okay. So we have to salute, this is his photograph, the great person of Professor Anand Mohan Chakrabarty for his great contribution to the scientific community for the creation of superbug. And how he created this very interesting experiment, okay. This is the overview of the experiment, okay. As I said, strain A of Pseudomonas putida, that has got a CAM plasmid, okay. Strain B of Pseudomonas putida, CAM plasmid will be able to, because of the presence of this plasmid, okay, they are having ability to degrade camphor. Then second strain B, that has got octoplasmid, and because of this particular plasmid they are able to degrade octane, okay. Then the strain C of Pseudomonas putida, they are having xyl plasmid, because of the presence of xyl plasmid they are able to degrade xylene, okay. Then the next one is NaH plasmid, that NaH plasmid, because of the presence of that particular plasmid, okay, they are able to degrade naphthalene, okay. So strain A, B, C and D. Then what he has done is, first he Pseudomonas is a, that they have a mating by using conjugation, okay, by using the pile, this plasmid, okay. The plasmid they are they had the mating of this particular bacteria, okay. These two strains resulted a recombination, okay. This will be half of the plasmid is going and integrated with the this particular plasmid, and this one is combined and they become a plasmid. And this particular plasmid, that is strain E, that has got a camphor and octane plasmid. And what he noticed is this bacteria could degrade both camphor and octane, okay. The beautiful study. Similarly, he has identified strain C and strain D, okay. This again he conjugated the strain C with the strain D. Then what is happened is here homologous recombination happened, but here it did not recombinate. Recombination did not happen. So what has happened is both this particular plasmid is simply went and transferred and coexisted together, okay. Along with this NaH plasmid, xyl plasmid also coexisted, okay. So this strain F has got two plasmid because of the presence of two plasmids, okay. So this also has got this also has got the ability to degrade xylene and naphthalene, okay. Now this strain E and strain F, again he combines together. He has undergone this conjugation, he has mated this particular thing. Then the result was very interesting. And this homologous plasmid that has both the CAM and octoplasmid went under a new bacteria to transfer to a new strain, okay. They transfer to a new strain. There you can see a CAMP region that is shown in the blue color and oct region that is shown in the yellow color, and these two are like the previous thing, they simply went and coexisted, okay. The xyl plasmid that is shown in the green color, and the NaH plasmid that is shown in magenta color, okay. So there are 4 gene this particular bacteria has got, okay. And what has happened? So individually all bacteria could degrade one plasmid, and this is the high super power bacteria, oil eating bacteria. They have got a CAM, oct, xyl, NAF gene. Because of this one, a single bacteria could degrade all 4 hydrocarbon. It was a great surprise, okay. That is what I told. We have to salute the contribution of an Indo-American, and this is basically, okay, an Indian scientist, okay. So that is the contribution of Anand Mohan Chakrabarti. So this is all about how there are a lot of further research are already happening. So this particular things I have taken from a beautiful textbook, Molecular Biotechnology. Now the excellent textbook called Glick and Pasternak. Okay, so you will be able to see in our library, that is available. Okay, so that I have taken all this particular details. So this is very interesting concepts. Okay, how Chakrabarti created superbug, they are able to degrade crude oil, and this particular Pseudomonas putida is known as oil eating bacteria, or degradation in oil spills. Okay, so this is all about the hydrocarbon degradation. Then today we can summarize the class. We have discussed one of the important topic, the degradation of microbial degradation of petroleum hydrocarbons, introduction on petroleum hydrocarbons, classification of petroleum hydrocarbon we have discussed, aliphatic and aromatic. Then the different classes of hydrocarbon in crude oil. Okay, that is aliphatic, aromatic, and cyclic. Okay, so that is we have discussed. Okay, then you can see that the major approaches for the petroleum hydrocarbon degradation. Okay, we have discussed the physical method, chemical method, and biological method. More accurately, biological method. Then what are the general factors which influences the this particular process, and what are the specific factors, environmental factors and microbial factors, then the hydrocarbon factors. Okay, specific factors that is influencing the hydrocarbon degradation. Then the microbial degradation of petroleum hydrocarbon, especially aerobic degradation, there is a role of oxygenase enzymes. Okay, and what are the major enzymes? Monoxygenase, dioxygenase, okay, hydroxylase, okay, all those enzymes. Okay, and the microbial enzyme in petroleum hydrocarbon degradation, how it is degrading both aliphatic hydrocarbons and aromatic hydrocarbon, and the end products are non-toxic components. And what is the role of biosurfactant? Okay, recent studies revealed that the hydrocarbon degradation will be influenced by the presence of a biosurfactant that is produced by the bacteria. Okay, like rhamnolipids. Then the limitation of all this problem. What are the conventional limitations? Okay, and for that particular limitation, amazing development, an amazing discovery made by Ananda Mohan Chakrabarti. Okay, Ananda Mohan Chakrabarti. So he has created the role of what is the role of genetic engineering? Okay, recombinant DNA technology. He transferred multiple plasmid that has got various capability to degrade various hydrocarbons into a single strain of Pseudomonas putida, and he constructed a superbug. They are able to degrade 4 different hydrocarbons. Okay, xylene, naphthalene, octane, and camphor. Okay, this is all about today's class. Okay, so these are the textbook, and one important textbook I told that is not mentioned here. Okay, Molecular Biotechnology by Glick and Pasternak. Okay, that is very good textbook. Okay, that you can see. Okay, and whatever I have told, especially for petroleum hydrocarbon, the textbook is very good. Okay, that you can refer. Okay, so thank you very much for listening to the class. If you are having any clarification, you can ask me. Okay, so this is very important topic, so we should listen carefully, should understand, especially Chakrabarti's experiment. It is very important. Okay, so thank you once again. Have a great time.